coming to our last science talk of 2023. I want to remind you that this initiative is developed in collaboration with the Environment Agency Abu Dhabi, Emirates Natural History Group Abu Dhabi, the Natural History Museum Abu Dhabi, and Nautica Environmental Associates. And I just want to tell you as well that we are in the preparation of the program for 2024. So I know already that it will be very exciting. So keep in touch and you will see the program coming up pretty quickly. Tonight, we will be talking about one of my favorite marine animals. And if you know me, you know that I have a penchant for underdog species. My background is plankton ecology, so yes, I do like little, and I do like eccentrics. Well, the animal we will be talking about tonight are certainly a special character, cuttlefish. And I'm wondering what comes to your head when you hear this word, cuttlefish. For me, first souvenir, it's walking along the French shores to collect cuttle bones to give to our hens at home. So not much interest in the animal itself, actually. And then the marine biology classes at university and the lessons on cephalopods. That was mind-blowing. It was about that time that I really had my first encounter with individuals underwater. And the charm operated. So I'm hoping that they will have the same effect on you tonight once you'll discover more astonishing facts about cuttlefish. And tonight, we have our researcher, Dr. Alexandra Alves, coming to stage talking about the biology and the ecology of cuttlefish. And then we will have a special guest from France, online, Dr. Antoine Minet, who will be presenting some results of his PhD regarding the impact of ocean acidification on cuttlefish behavior. So stay here, this will be very interesting. And the last speaker, and I lost her, oh, she's here. This would be, Anupama Chaudhary, who will be presenting our Science Citizen Initiative. And this would be super exciting as well. Just a reminder, keep all your questions for the end. We will have the Q&A question at the end. So we will go through the three presentations and then a Q&A session at the end. All right. I will welcome Dr. Alexandra Alves now. And I'm very happy to call her on stage. Dr. Alex has joined the center from Portugal, where she undertook her PhD studies focusing on fish physiological adaptation to ocean acidification, here again. Dr. Alex is an animal physiologist with expertise in neuroscience, aquaculture, physiology, and climate change topics. Here in the center, Alex focuses on the cuttlefish culture and the mitigation of bottleneck encountered in the aquaculture industry. But she's also aiming to better understand the physiological adaptation of the local cuttlefish species to the natural environmental stressors. So welcome on stage and have fun.
Good night, everybody. Thank you for coming. So today we are going to talk about cuttlefish. Cuttlefish is not a fish. It's a mollusk of the class of the cephalopods. In this class of animals, there are three superorders, the Nautilidae, the octopodiforms, and the decapodiforms. These orders can be distinguished by animal features like the shells, the arms, the eyes, and the beaks. So let's talk a little bit about them. Nautilidae are unique class of cephalopods, being the only order that has still an external shell. The modern-day nautilus make up the only extant order that have remained relatively unchanged for millions of years. That's why they look alike so much as the fossils. The members of the octopodiforms are the octopus. During the evolution of this species, a erasure of the mollusk shell trend gradually appeared, which is already absent in most of the species. In the decapodiforms, we have two orders, the Teutidae, that is composed by the squids, and the Sepidae, that is composed by the cuttlefish. The Teotidae order is the most diverse order of the cephalopods with more than 300 species. Like the octopus, they evolutionary been losing their shell, being absent in most of species. But that's not what happened with cuttlefish. Cuttlefish have the most mollusk shell within their bodies and is known as cuttlebone. We also can distinguish the cephalopods for the amounts of arms and tentacles that they have. Nautilus have more than 19 tentacles with no suckers. Octopus have, how the name indicates, have 8 arms with usually 2 rows of suckers, and these suckers have cavities to ensure the tight grip on prey. Both orders of the decapodiforms have 8 arms and 2 tentacles, making a total of 10. The squids have clubbed ends on the tentacles composed of stalked suckers used for hunting. The arms have 2 to 4 rows of suckers, which enable a stronger grip while feeding. The cuttlefish, the arms are shorter than the tentacles. The tentacles have large, densely stalked suckers, while the arms are often only with 4 rows of suckers. The 2 tentacles are retractable. Here are some images of cuttlefish using them to capture prey. As you can see, the tentacles are really fast, coming out and retracting. What about the eyes? Nautilus has a simple pinhole eye and then don't have good vision. Octopus have single-chambered eyes. Squids have the largest eye-to-eye body size ratios of all the animal kingdom that can measure up to 24 centimeters. Cuttlefish have unique eyes, and the pupils are often shaped like a U or a W. Surprisingly, they are colorblind, but we are going to talk about that later. Now that we already know how to distinguish our cephalopods, let's focus on the cuttlefish. Cuttlefish have a closed vascular system that distributes the oxygen from the gills to the rest of the body. They have, in their circulatory system, they have 3 hearts, one systemic one, which distributes the oxygenated, rich blood into the rest of the body, and 2 branchial ones, which receive the oxygen per venous blood and create the pressure to pump it through the gills. And we can see it here. This is one of the branchial hearts connected to the gill. Another interesting fact about these animals is that their blood can be blue or green when it's oxygenated and colorless when it's deoxygenated. The reason is that they don't have red blood cells like us humans. As humans, we have iron-based pigment called hemoglobin that is used in the red blood cells to transport the oxygen. The cuttlefish have a copper-based pigment named hemocyanin that is freely dissolved in the blood. How do these animals move and hover in the waters? The cuttlebones, the mollusk shell within the cuttlefish bodies, control buoyancy. The cuttlebone has 2 main structures: the dorsal shield that provides the stiffness and acts as the base for the lamella matrix that is full of chambers having high porosity and high permeability. In this porous material, cuttlefish can control how much liquid enters the cuttlebone. So the ratio between the gas to liquid inside is determined by the depth they want to go. More gas in the cuttlebone means they will float. More liquid means they will sink, making it easier for these animals to be neutrally buoyant, hovering in the waters. To swim, they use the fins that are all around the mantle and moving like waves. When they want to be fast, they jet propose water through a siphon that is the hole that you can find under the arms here. They also can jet ink to hide themselves from predators. Inking gives them time to escape, creating animal forms that are named pseudomorphs, confusing the contour-specifics of predators about their whereabouts. There are also small cuttlefish species, as the flamboyant cuttlefish, that as an adult only reaches 8 centimeters, And these animals, because they are small, they also have small cuttlebones, which make them inefficient control buoyancy. So they use another technique in locomotion named impaling, which means that they use several arms and fins as leg stumps to slowly move across the seafloor. Cuttlefish have the ability to change its appearance, a technique that they use to hide from the predators, to ambush prey, and even to fool counterspecifics. To make this change in appearance, they use several elements. The chromatic, that is not only the colors, but also the patterns, like stripes, spots, and bands. For example, some displays is to frighten the predators, because normally exuberant colors in the ocean means that something is poisonous or venomous. The two black dots in the back of the mantle is used to mislead the position of the head to threats, because the position of the head on the body normally indicates in which direction the animal will escape. The zebra display indicates to the females that the male is ready to breed, and between the males is used as a display of the competition to mate with a female. But we also have other different elements, like the textural element that is related to the smoothness of the skin or the population of the skin. We also have the postural element that is related to the position of the body parts, like the attitude of the arms here, or the spread of the mantle. And finally, we had the locomotor that is about the typical movements, if they are resting, if they are burying themselves, if they are walking, if they are inking, and so on. So now that you know the elements of camouflage, can you spot the cuttlefishes? Some images are easy. So we can spot one here, one big one here, and one here. But other ones are harder. So you have one here, one here, here, and here. And here is an example of a cuttlefish ambushing prey. So I'm trying to just show it again. So he's perfectly camouflaged with the rest of the reef, and the prey doesn't see him coming. But how are they able to create these colorful patterns and textures? They can change colors in less than 200 milliseconds. In the skin, they have chromatophore organs by the millions. These organs have elastic sacs of color pigments that can extend by tiny muscles controlled by neurons. The chromatophores are generally only yellow, red, and brown, but you also already seen that the cuttlefish can display multiple colors. The shiny iridescence comes from the reflecting cells named iridophores. These cells are stacked on top of each other and reflect light in different wavelengths, making them appear any color that they want. So collectively, they allow the cuttlefish to have these amazing displays. So the combination of the chromatophore organs and the iridophore cells. The texture comes from a unique feature on cuttlefish skin named papillae that also are neurally controlled. The papillae are composed of erector muscles which leave spans in the skin with a variety of shapes. These shapes can match the surrounding 3D environment as the texture of algae, seagrass, corals, and rocks. But how can they camouflage and imitate their surroundings if they are colorblind? The scientists are still studying it. One of the hypotheses is that they can perceive color by chromatic aberration, which is a phenomenon where different colors come to focus on different distances due to the color wavelength bending or refracting through a lens. The W shape of the pupil lets light enter at different angles, enhancing the chromatic aberration phenomena. That, in theory, could help the cuttlefish separate colors by allowing them to detect differences in the blurring as focus changes. So they are not seeing different colors, they are perceiving differences in the levels of blurring and focus. Additionally, even in complete darkness, they can still camouflage with the surrounding environment. That is pretty cool. Cuttlefish also have peculiar behaviors. Let's talk about some of them. The broad club cuttlefish is a species specialized in hunting crabs in the reefs. It starts with sneaking up the prey using camouflage. But when it fails, this species starts a mesmerizing show. The cuttlefish uses the ability to change the colors and patterns with this specific hunting technique that is hypnotizing and is typical of these species. Cuttlefish are solitary animals, with the exception of breeding seasons. At this time, they can congregate together. Sometimes could be as much as thousands, like the giant cuttlefish in the coast of Australia. Let's focus a bit on the breeding behaviors. During breeding season, the males present this intense zebra display that are the white and black bands, and also discoloration in the forearms. The reproductive behavior presents different stages. The first stage is courtship, that is initiated by the male. During this stage, the male and female will swim in parallel. Male will hover over and alongside the female, continuously drawing his arms over her mantle, head, and arms. Then we have the mating. Eventually, male will grasp the female and copulates head to head. The males have a specialized arm that insert inside the pouch that females have under the mouth, releasing the spermatophores. The fertilization of the eggs is done externally. Each egg is held amidst the arms of the female, while spermatozoa are released from the seminal receptacle for fertilization. Males can accompany females during several hours needed for egg laying, which is named temporary mate guarding. The female wraps the stalk of each fertilized egg around some structure and dies shortly after spawning. And here we can see some videos of eggs found in the wild by divers. They always are attached to some kind of substrate. Each batch of eggs laid differs in number. It can be just a few, going up to 800 eggs in a single batch. That can take hours to lay. The color of the eggs differ between species. The Atlantic and Mediterranean cuttlefishes lay black eggs that are inked. And the species that habitat warm waters like here have white transparent eggs, probably to assist them in temperature regulation. In the aquaculture industry, we discern viable eggs from inviable eggs by color and shape. So if the egg color is not specific to the species, or it hasn't the perfect teardrop shape, they are discarded, because the studies shows they will have low hatching rates and a diminished survival of the newborns. What happens inside the eggs? Here you can observe the embryonic development of a cuttlefish. This development is temperature dependent. Generally, the increase of temperature will make the embryos develop faster, of course, within the range of the specifications of each species. The embryo cells forms on top of a large whorl that is used as an energy reserve. With the progression of the development stages, this whorl will reduce its size till it disappears, indicating that it's time to hatch. After more or less 10 days, the embryo starts to form organs like the eyes, the cuttlebone, the arms, and the ink sac. In 3 weeks, the chromatophores appear, and we start to see changes in the pigmentation. Over 4 weeks, what began as a tiny cell will develop in a hatchling that resembles a miniature version of an adult cuttlefish. When the cuttlefish hatch, they only have 1 to 2 centimeters, but they are already mini adults, with suckers ready to jet ink, to hunt, to display patterns, and to change the shape of the skin. In the wild, the survival rate of this hatchling is very low due to the predation and the insuccess rate of feeding. In the congregations of breeders of the giant cuttlefish, there is a lot of competition over the females. The larger males will put elaborate displays to intimidate other males. And it's common to observe fights escalating. So the smaller males have a technique to be successful in mating during this breeding season. They actually mimic the appearance and the posture of the females. This disguise fools the bigger males, and they are able to reach the protected females. And if they are lucky, they are also able to mate. These males are called sneaker males, and this behavior is observed in most of the cuttlefish species. You can see they are mating, and you have the big male protecting them. The mourning cuttlefish takes this technique further. When just one rival male is present, the sneaker male will split their colors and displays in half. In the side facing the female, it will display the normal male colors. And the side facing the rival male will display the female colors to not be perceived as a threat. I would love to go on and talk about cuttlefish. They have amazing memory. They are super smart. When I work with them, they can identify me, so they can distinguish people. They know who I am, who the keepers are, and who are the people that don't know. They can be trained. If you give them signals, they can come and eat from your hand. So I hope that now that you know a bit more about cuttlefish, you'll fall in love like I did.
Yes, I fell in love again. Thank you, Alex. Well, this was super interesting. All right. Time for our second talk, and I'm gonna call Antoine. Yes, Antoine is here.
Yes, can you hear me?
Yes, we can. All right, let me introduce you, and then it will be your turn. So I'm very honored, actually, to welcome Dr. Antoine Minet, because he's a fellow scientist from my home university in La Rochelle. He's carried out his PhD study at the University of La Rochelle in France, and his study was entitled Bioaccumulation and Effects of Mercury on the Cuttlefish in the Context of Ocean Acidification. Yes, this is a very interesting topic. So Dr. Minet has been working with cephalopods for over 5 years now, and is particularly interested in the effect of global change on the common cuttlefish, Sepia officinalis. But I'm gonna just stop now because he will tell us a lot more about his findings and his studies and his findings. Welcome on stage, Antoine, your turn.
Thank you for the introduction and thank you for the invitation. So I will try to share my screen and show you the presentation. Is it fine? Do you have the full screen? I guess so.
Yes, it's fine.
Perfect, thank you. So yes, so today I will talk a bit about my PhD results, so part of them, and we will focus on ocean acidification and the cuttlefish behavior of the common cuttlefish. So in the first 4 minutes, we will talk about a bit of the context of this study, in case you're not familiar with ocean acidification. And so we will talk about anthropogenic pressure. So We first studied anthropogenic pressure because in the 19th century, we had the first industrial revolution. We came from end production methods to machines, so we knew nice economic growth, but also we increased pollution. And it's only by the end of the 20th century that we start talking about global change. So we start talking about habitat destruction, climate change, pollution, but also ocean acidification. This latter is well known because it's due to the high CO2 emission we have from industry, transportation, and so on, which finally will dissolve into the water, react with the water, and form what we call carbonic acids here, and also hydrogen ions. These hydrogen ions will increase what we call increase or decrease the potential hydrogen, which is also known as pH. So if we look to the scenarios of the Intergovernmental Panel working on the climate change, they estimate that the pH of the ocean, which is now about 8.1, will decrease on about 7.65 by the end of the century if we take the worst case scenario. In fact, it's a logarithm scale, so it means that the ocean will be 6 times more acidic than right now. We already know some effects of ocean acidification, for example, on calcification. Here's a short example of pictures of corals, which are dying and finally totally dead in less than a year. But there is also some impact on embryonic development, immunity, but the less known impact is about neurotoxicity. In fact, the excess of CO2 and production of H+ shows that neurotoxic effect on different animals. To study this neurotoxicity, we choose to work on the common cuttlefish for several reasons. First, because it's a major fishery resource in France, but also in a lot of countries. Then it has a short life cycle, 1 to 2 years, so grow fast and die young system, so with a high physiological performances which can be disturbed by this ocean acidification. Then it's a semalparous species, which means that adults will give birth only one time in their life, and then they will die. So the cuttlefish will not have any parental care. So they will have and need complex behavior to be able to survive in a hostile environment. So for us, it's a really good model for what we call ecotoxicological studies, and particularly for behavioral studies. So today, what do we know about ocean acidification and the effect it can have on, for example, fish? We know that it impairs motor function, olfaction, and predator avoidance. If we go and move on only cephalopods, we know it alters defensive and predatory behavior in the pygmy squids. However, if you go to other cephalopod species, there is no data at all. And so that's why we decided to go further and to study what happens on the cuttlefish if we expose the cuttlefish to lower pH. So our hypothesis was that ocean acidification will, of course, affect fundamental behavior of animals. So let's move on what we found in the lab. So first of all, I would like to detail you a bit about the exposure. So we sampled some eggs in the port of La Rochelle in France, and then we divided them in 2 groups. One group will be exposed to the ambient conditions, so the control condition here in green with a pH of 808, and then we expose them to acidified condition with CO2, pH of 7.54, which will be a pH that is really close to what we can find, we will find by the end of the century in coastal environments. So they were exposed for about 1 month, and then when they spawn, we had different groups. So we kept 54 animals, and then for 1 month and a half, we conducted every day some behavioral assays. So here is a small resume of what kind of assays we've done on the cuttlefish. So predation, locomotor activity, eye preference for vigilant scanning, background matching, which is part of the camouflage, predator detection, and then we also check the visual acuity. But for today, we will only focus on 4 types of assays because we don't have all the evening. But if you have any question for all the other assays, we have the Q&A for this. So we focus on defensive behavior because it's a really important behavior for the survival. And usually, as for most animals, the defensive behavior are divided in two groups: primary defenses, which finally are composed of vigilant scanning, background matching, masquerade, mimicry, and freezing. But Alexandra already showed you some pictures. These primary behavior defenses are just to avoid detection of the predator, and then if they are detected, they will use the secondary defenses, which are summed up in intimidating, flight, burying, and inking. As you can see, it's a really rich and complex behavior, and it will directly impact the survival of our animals. For today, we will not only focus on 3 primary defenses and more on all the secondary defenses. So first of all, have you seen the camouflage can take different shape and pattern, and we will only focus on disruptive patterning, which finally the goal is to break up the optical appearance using highly heterogeneous pattern and contrast. So what we do is we put the cuttlefish in a checkboard pattern and look at different what we call chromatic components, so 11 of them, and then we look if they are highly expressed or not expressed. So highly expressed, it's a score of 3, not expressed is 0. So the disruptive score at the end is between 0 and 33. 33, it's a highly disruptive cuttlefish, and close to 0, it's a non-disruptive cuttlefish. So here you've got some of the chromatic components you can see here, and you can report if you want on the cuttlefish to identify them. So here are the results we found in lab. So you've got the disruptive score here from 0 to 33, and here you got the 2 condition with the control condition, and then the acidified condition with the CO2. The box you can see here represents most of the individual, and as you can see in the control condition, most of the individual are highly disruptive. So they are just trying to match the background. So that's really good. But in the acidified condition, most of the cuttlefish did not match with the background. If we want to look at what happened into picture, here's a classical control cuttlefish, which will have something like 27/33 in the disruptive score, while in the acidified condition, we are really close to finally 0. So we have a strong effect of pH after 1 month of exposure. But the question here behind that is, does the cuttlefish cannot or will not use disruptive patterning? Is it a question of perception or willingness? So to check part of this question, we decided to verify the visual acuity of the animal using an optomotor response test. Optomotor response is a reflex involving the turning of the head or the body in response to moving stripes for the animals. For example, in her case, it might be like if I clap my hands really quickly and fast and strongly, like you will close your eyes as a reflex. It's the same thing. So if the animal is seeing properly, the animal will turn and follow the stripes. So here you've got a little scheme of the animals into the aquarium, and here are the moving stripes. And we record this with a camera. Here is a short extract. So you've got the little cuttlefish here in the middle. And we are moving the stripes around. And as you can see, the cuttlefish is taking one stripe and following the stripe, meaning that the cuttlefish can see properly. So we repeat this operation in 3 speeds, 2 direction, and without strikes to be sure that it's not random and the cuttlefish can really properly see. So we have no effect on pH, meaning that the cuttlefish do not use the disruptive patterning, but not because they cannot see it, but maybe because they cannot, they don't want it, or they cannot because there is something else which is blocking neurotransmission in the brain. These results are also confirmed with the predation, because most of the predation, as you've seen previously, are led by vision, and we have no problem or no alteration of the predation in acidified condition. Then let's move on high preference for vigilant scanning. High preference is laterality. It's like, for example, in human hand preference, if you're right-handed or left-handed, means that you prefer to use some part of your brain for some skills and another part of your brain for some others. Usually, we know that in cuttlefish, the left eye is used for anti-predation, like shelter seeking, vigilant scanning, while the right eye is used for predation, prey attack. This gives a really nice advantage because they can do what we call the dual tasking, so they can hunt and stay vigilant at the same time. So to do this test, we put the cuttlefish in a new environment. Usually it's something really white with a lot of light on which they don't feel really confident and they are a bit stressed. So they really stay vigilant for a long time. So we put them in this kind of tank for 90 minutes, and every 5 minutes we will look at with which eye they will look at the surrounding, because they will usually stick to one of the to a side of the tank, and then we will look with which eyes they will use. So for example, if we take this cuttlefish, the cuttlefish look with the left eye and this one with the right eye. We develop what we call the laterality index, but I won't go in too much into detail. What you have to keep in mind is only that if the cuttlefish use the left eye, you will have green on the graph. If it looks with the right eye, you will have red. So here are the results we've got in the lab. So the cuttlefish eye preference in percentage for the control condition and the acidified condition. What we've seen is that in the control condition, most of the cuttlefish, so about 80%, prefer to use the left eye to look at for the vigilant scanning, sorry. So in less than 20% the right eye. When you move to the acidified condition, it's about half and half. So usually you've got a majority and you have what we call a populational level laterality. In our case, in acidified condition, we have an individual level laterality. So our question behind that was, okay, does it have any consequences on the development of the brain? Does the cuttlefish will answer as fast as the others and with the same reaction in case of predator? So we move to the next test about predator detection. So in case of vigilant scanning, if you detect a predator, then you can choose to do not have a behavioral response or to have a behavioral response. In this kind of response, you have the body patterning, which is a change in brightness, pattern, papillae, as you've seen previously, for example, here on the top, and a change of arms position. Then you've got also some deimatic response with acute disruptive or conflict model, for example, the two black spots you've got there. And you have also what we call the protean response with the flight and the inking. So here those reactions are classified by intensity, with body patterning being the less intense and then the protean response being the most intense response. So what we've done is that we took an iPad. On this iPad, we put a looming stimuli, which was a shark shadow, which appears and come really close to the cuttlefish. So you've got the cuttlefish here in the aquarium, and we add also a mirror at 45 degrees, which gave us a view of the side of the aquarium and also the top of the aquarium in the camera. So we looked at the detection latency of the cuttlefish to answer, to react, the type of reaction, and then the visual field is used to vigilant scanning and to react. So it's a short movie. You can see the shark on the screen. Here you've got the cuttlefish, the shark, and if you look, two black spots will appear. So that's why what we've seen previously, a deimatic response. If we change the cuttlefish, so here the shark will come by the other side, and the cuttlefish is doing with the two eye. We have another type of response, which is the protean response, the more intense response with a heavy inking. So quickly on the results, we've seen no effect of pH on detection latency, but what we saw is that about 20% of the cuttlefish exposed to low pH had more intense response. They have the tendency to use ink more frequently. So our question was, is there a neurotransmitter alteration? Is there a problem of inhibition of some neurotransmitter? And so do we have a more anxious individual? Because finally, it's what in the literature which seems to be going on for this animal exposed to acidified conditions. So if I have to sum up what we've seen previously, ocean acidification alters cuttlefish behavior, so camouflage, predator detection, lateralization, and so on, but we still don't know exactly mechanistically how. Also, we could do more realistic approach and keep in mind that cuttlefish are not only exposed to ocean acidification, but to multiple pressures, like some other pollutions, a change in temperature, and so on. And there is also some dynamics. So here you've got a graph of the dynamics of the pH with the day and light fluctuation. And then we can also wonder if there is some reversibility. If, for example, the pH going back to normal, do we still have the effect? So it's a high complexity system. Finally, if we want to be really close to, I would say, environmental condition, but it's worse. It's worse. And then we can also wonder if cuttlefish will be able to acclimate and to have some local adaptation to the condition. Thank you for your attention. So if you have some question during the Q&A, do not hesitate. And if it's after, you have my email just right here. Thank you.
I have a question about the shark. Did you create that shark especially for the experiments, or I mean?
Yeah.
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 You can answer after, or you can answer
okay.
now. Go ahead. Go ahead. Go on, answer. I asked the question.
Okay. In fact, there is some Australian which did this with some cuttlefish also once. And they used different predators like shark, like big fish, and they also tried with some crabs. So we did the same, and the one with most intense response was the shark. And as we wanted to see some difference, we took the most intense response. So yeah, we just took what was already available in the literature.
That's pretty good. All right, thank you. All right. Third speaker, Anupama Chaudhary is here, and so I will welcome her on stage. Anupama joins SeaWorld education team in 2022. She has a master's degree in GIS, and you will see that it's quite important for what she will be talking about. Right now, she's very keen on environmental conservation, and she has worked with environmental outreach and education project with the UAE government since 2013 already, with a special interest in blue carbon ecosystem. And just to say that she will be presenting our citizen science initiative, but it's not the first citizen science initiative that she's been developing and working on. So welcome on stage, Anu, and very excited to launch that citizen science initiative.
Just a second. Okay. Yeah, it's here. I'll just present. Okay, thank you. All right. Hi, everyone. I'm just going to start with some images that have been shared with us. These photos are of cuttlefish from Solnhofen, Germany, and these fossils are around 152 million years old. So cuttlefish have been around for a long, long time. Okay. Okay, so my name is Anu, and I'm here to talk to you about citizen science initiative that we are undertaking right now, and how you can get involved in this project. And as members of the community living in this beautiful coastal country, you are our most valuable resource in this research that is happening. So before we move forward, can I ask you guys, how many of you here go to the ocean pretty frequently for boating, fishing, diving? Can we see? Oh, nice. That's awesome. Okay, great. Awesome. All right, let's move forward. So these are some of the components of a citizen science initiative. So you can see the many different things that are involved here. And the simplest definition of a citizen science initiative or a project is when the community voluntarily helps conduct scientific research. It can strengthen the social bonds that make a community resilient, which is the most important element that we are expecting to combat climate change, is a resilient environment and a resilient community. So it also gives people an inside look on how science actually works. So getting involved in a citizen science project helps generate new knowledge or understanding to tackle environmental concerns around us. For this project, we are looking at cuttlefish, and we are going to be helping Dr. Zana's cuttlefish research. So let me tell you what is the aim of this project that we have here. Basically, to collect data from a research study on the populations around UAE. So it can be around Abu Dhabi, Dubai, anywhere, any coast in UAE. And to raise awareness on cuttlefish and their habitats. The Gulf's marine ecosystems are obviously very complex and and there's a lot more scope for studies going on. And so that's why it is important to know about the marine biodiversity and help our next generation to understand the marine biodiversity as well. So citizen science projects can be utilized for many different types of data collection. So when the public gets involved, there can be places where professional scientists can't really go all the time, right? So it can be about recording earthquake tremors or observing honeybees or counting meteors, ladybugs, or birds. And in this case, we are talking about the cuttlefish when you go diving in the ocean or when you go close to the ocean. So the data collected here will increase our knowledge on the species in the Gulf, and it will be completed with laboratory studies here at SeaWorld Research and Rescue. So how do you get involved? So who here likes to go to the ocean? Nice, that's awesome. So when you go to the ocean next time, when you go to the sea, when you go there, generally you go by a boat. And when you're in a boat, most of the time, the boat has a GPS system, right? In this day, it is highly unlikely that the boat would not have a GPS or a location. So the most important part that you have to remember if you would like to take part in this initiative would be to record the location of where you spotted the cuttlefish. So when we go in the ocean, you're looking at lots of different things. Cuttlefish are generally not a very glamorous species, but we tend to look for other bigger species like the turtles or the dolphins or dugongs maybe. But if you see any cuttlefish, please make sure that you have it in the dive log or even in the just note it down at some point of time. We would love to see pictures as well. So if you could take any pictures, if you have underwater cameras that you take along with you guys, it would help us immensely and to see what kind of environment they basically live in, as well as many other information that we could collect from it. When you spot a cuttlefish, I would like to tell you, if you would like to take part in this initiative, make sure to observe, look at it, look at its colors. You can see where it is, where you have seen the fish. Fish, I called it a fish. Where you have seen the cuttlefish, as well as what kind of environment was it in. Let me show you the form as well, so you will get a better idea of what kind of data we are looking to collect. If you're unable to, if you do not have a GPS at that exact point of time, but if you have a phone with you, you can easily collect the location coordinates. Just open Google Maps and you can see where you are. And if you click on that point, you'll be able to see the location coordinates. And once you have that, that's the location that we require. That's it, right? Okay. Okay. Let me open this website. Okay. This is our website for the Cuttlefish Citizen Science Initiative. And when you come here, we have this Begin Questionnaire button. You can click on this questionnaire, and it will take you to the form where you can input all the information that you need to do. So we would love to have your name, your email ID, and which emirate did you spot the cuttlefish in. Date and time would be great. And if you have an area, so I know there are many diving spots around here that we call the wreck, or we have those cement blocks somewhere around Abu Dhabi. So if you know the area roughly, that would be perfect. And we have given examples for what latitude and longitude numbers look like, so it won't be confusing. You can input those in here. This is a pretty simplistic form. We don't want you to overwhelm you. If you want to participate, it can be easy to fill it up, and you can do it later on. We just need to make sure you had the location coordinates for this. After that, we have the species sighting. So if you saw a cuttlefish, or if you saw any octopus or any other cephalopod, squids. Confidence level. So it's we obviously understand that when you are underwater, sometimes you might see something and you think it might be a cuttlefish, maybe not. So we have the confidence level here. So hopefully that will help us and help you as well. We would love to know where you observed it from, because I have seen cuttlefish from the Eastern Corniche, just walking on the walking on the promenade there. I have seen cuttlefish in the canal right there, near the Eastern mangroves. So we would love to know where you saw them from, if you were in a boat or on the beach or anywhere else. This is the physical description of the animal. We would love to know if you thought the animal looked healthy, or it looked like it needed some care, or if there was anything specific that you thought about it. Generally, we have observed that if you see one cuttlefish, there might be a few of them around somewhere. So if you know the number, if you can see the number, that would be perfect. Here you can describe what it was doing. So was it trying to feed, or was it trying to hunt an animal? Was it just chilling around, or was it near any eggs? That would be very helpful. If you look at the animal and if you see any plastic around it, we would love to have that information, because that would give rise to other questions, and that would be a part of the research. Seabed type. We have given a drop down here, so you can put in whatever kind of seabed you saw the cuttlefish in. And eggs. If you do spot eggs, that would be wonderful. Please record them. And it's obviously very difficult to count the number of eggs when you're underwater, but if you can write an estimate, that would be wonderful. The eggs are generally attached to a substrate. Like you saw in Dr. Alexandra's research, the eggs were on a coconut shell. If you can write some kind of description of that sort, perfect. And we've seen cuttlefish bones everywhere on the beaches where we go in UAE, right? So if you spot any cuttlefish bones, please use this form. That would be wonderful for our research, and we can have that information. Dr. Alex has been kind enough to give us some sample of how to take a picture if you spot a cuttlefish bone. We always need a scale. So a banknote or a coin would be an ideal scale because everyone has that, and that gives you the exact size, that gives you an estimate of the exact size of the bone. We have the location here as well. And if you feel like you saw something very noteworthy, like anything that you think was out of the ordinary, you've seen many cuttlefish by now, and you saw that this was not something that I saw the other cuttlefishes do, please note that down as well. That would be wonderful, so we can figure out the behavior. So I was told to put in a joke in my presentation, so let me tell you there's nothing fishy about this page. We have to put in this we have to put in this capture image here for security reasons. So here it goes. And then you just submit it. And then after you submit it, we'll get that data, and we're going to put the locations on a map, and that map will go on this website. So you'll be able to see your inputs on this website as well. So it would be nice to see lots of records coming in, and whoever's diving or going swimming or to the beaches, I would love to see that you're recording the cuttlefish records. All right, thank you so much. Okay, so if you would like to contribute to this project, let me show you the QR code really quickly. Sorry. Sorry, just a second. Yeah. Here's the QR code.
Great. Thanks, Anu. All right, Q&A session time. Come back on stage, and we can have Antoine back on screen. The microphones.
Hi, for Dr. Alex. How many species of cuttlefish do we have in the Gulf?
Here in UAE water?
UAE, yeah.
They are described 11, and 3 of them we only can see on the Fujairah side.
Okay, thank you.
All right, yes, Ali?
Yeah, I'm curious about the inking. How many times can a cuttlefish ink?
A bunch of times. Yeah, because when they are inside the sacks, it's like a dry pigment. Then when it mixes with the water, it's like the huge quantity that you see.
Hi, sorry, you mentioned that look out for signs of when a cuttlefish looks unhealthy. Is there anything in particular that is very obvious for people who don't know a lot?
Normally, when they are like whitish, it's because they are dying. So.
All right.
Antoine, do you want to add anything to that?
Yeah, I think we can also, like, if they are floating too much also, and it happens depending of the reproduction. For example, I know on the coast in France, you will have a period on which the male will die, like it might be around, let's say, May, and then you will have the female a bit later, like in June. And usually you can see them around, and they are not going on the floor anymore, just floating like that. And yeah, that's a crucial parameter, and which is really hard to. And then, let's say, which will modify the statistic, like most of the individual you will easily see, will be the individual which are dying since they are not using camouflage anymore and floating. So that's why it's a key parameter, I think, to just report and to say if they are healthy or not at the moment where you are seeing them.
Great, thanks. Another question? Here.
You mentioned about how cuttlefish are affected by rising ocean acidification. Are they affected, like, what's their current population?
Sorry, say that again?
What's their current population?
What's their current population? It's a good question. Antoine.
Yeah, that's really hard to evaluate the stocks of cuttlefish. Yeah, because they are moving and we don't know. The stocks are renewed, in fact, every year because it's a short life cycle of cuttlefish. And sometimes you're like fishing and doing, let's say, an evaluation on the coast, and you're like, okay, there's only, let's say, 3 or 5 tons over there. And then 2 years after, you have 10 times more cuttlefish because they are moving and going where they have some food, avoiding fishermen and so on. So it's really hard to evaluate how many cuttlefish, in fact, there is in our coast or even in all the oceans. I cannot answer this, but yeah, probably millions, but then I don't know the exact number.
Like, are they affected by anything else like microplastic or ocean levels rising?
Yeah, of course. They are affected by temperature. Then you can find microplastic in their tissues also. You can find also a lot of heavy metals. So during my PhD, I found a lot of mercury in the cuttlefish because they are really active animals when they are, let's say, chasing for some food, and they are eating a lot compared to some fish. They can eat something like 20 to 30% of their weight, so just per day. So just imagine for you eating 20 to 30% of your weight. So they really accumulate a lot of mercury, and then if you're eating the cuttlefish, you're eating also the mercury, the microplastics, and so on for your turn. So that's why it was a crucial question. But yeah, you have a lot of also pollutants in the cuttlefish, and not only ocean acidification, which is a threat. Yeah.
All right, thanks. A question over there. Yes.
Thank you. I'm going to be cheeky and try and sneak in 2 questions. The hemocyanin, which you mentioned in terms of cuttlefish blood, and we see that also in things like limulus, the zifasurin. Now, those are bled for their blood yearly because it's used by the pharmaceutical industry. Has the same been done for cuttlefish yet?
Well, because normally the cuttlefish are small, not the giant cuttlefish can grow up to 10 kilos, but normally the other ones are smaller. That's why they are not used to extract the blood, because it'll be like a small amount.
Okay, that's interesting. And on the mercury, why do cephalopods hyperaccumulate mercury? Because it's not just pollution, they do it naturally. We've been studying cephalopods from the Jurassic and the Cretaceous, and when we've scanned them at the synchrotron, we've found high concentrations of mercury within the gills, and there was no pollutants around then. So why would they hyperaccumulate mercury?
Maybe for you.
That's a good, yeah, that's a good question. There's different reasons. Like mercury, let's say like 1/3 to approximately 1/2 of the mercury is natural because it's coming from volcanic sources, and then the other part is due to human activities. So in their muscle, they have a lot of proteins with sulfurhydride groups, which are really likely, let's say, friends with mercury and will bound really strongly with the mercury. And since they are not that much, they are close to the top of the trophic chain. Finally, if you consider the big cuttlefish, the adult cuttlefish, they can go to 10 kilos, and they will eat pretty much big fish or big crabs. They will accumulate a lot of mercury because they are, let's say, really close to the top of the trophic chain. So it's common to see mercury in let's say, every fish, even in every fish and every cuttlefish also.
Thank you. All right. Claudia, sorry, I didn't look. We'll come to this side after.
Yeah, I have a question. Is the fishing of cuttlefish in numbers restricted here in the UAE or not? Are there any controls on the fish markets? How many cuttlefish are there? And I guess a lot of private people are fishing as well cuttlefish. And when we observe them, they are pretty often close to the coast, so they might be really easy to catch. Yeah, so the question is, how can the numbers here be controlled?
Anu, do you have any insights on the regulation? I don't know.
The regulations also don't know, but when I was doing questionnaires with like fishing expeditions, normally they don't target cuttlefish here in these waters. And then when I went to the market and asked where the cuttlefish came from, most of them came from Oman, so not UAE waters. But that's just questionnaires that I did.
I guess the answer is that nobody really cares about cuttlefish right now in the Gulf. So it's knowing the status of the population, knowing the numbers, and if they are affected by fisheries, it's not known.
Yeah, but it would be interesting also to check, because we have a lot of dredging and artificial islands here. So how does this influence the cuttlefish population? I'm not a diver, and I just saw in 3 years 3 cuttlefish close to the coast, and I think this is not a very high number. And so the question is really, how many cuttlefish do we have here? And I hope you can find it somehow out.
Well, our initiative will help us to do that. That's part of one of the objectives of the initiative, is to find out how our population is doing here.
Really good questions. Hello. We need a local voice here, from my experience. Cuttlefish here, we call them khathag, to differentiate them from regular squid, nagar. And khathag, usually, the way we catch them, we harpoon them on the coast, because they're easy to harpoon. And we only target the Faro cuttlefish, because it gets to be this big, and it can be used in a stew, for example, for a family. It doesn't have a very big commercial importance, but some families use it as a resource for food. But it's a known dish here. I wanted to share an experience I had when I was a kid. I was walking with my mom and my sisters by the Corniche before it became this sandy beach. We had all the rocks and rock pools, and you can see the corals and the angelfish that were breeding over there. Sometimes you have low tide, and you can see a lot of debris. And we found this burlap sack. It's like a flour sack or something. And we just lift it, and we see this small bundle of white grapes, okay, which were eggs. I didn't know what they were at the time. We were little kids. And we took them, we put them in a plastic container, and we filled it with salt water. Okay? And I go back to my books, and there was a book by Frances Dipper. She was here in the UAE. But I managed to get her book on marine and freshwater wildlife in Britain. And there was one page on cuttlefish and the cuttlefish cycle, and I identified the eggs. I'm like, Mom, we have cuttlefish eggs. And here the thing is, some of where the bundles were kept together, they were white. But the ones that separated started to turn yellow. And then I found out the ones that turned yellow won't hatch. So we tried to not separate them. Just took a few days, and we can see something inside, these tiny ghosts with these small tentacles. And then they emerge. And instantly, the moment they come out, they can change color from white to dotted to red. And they spew this tiny dot of ink. The moment they were hatched, they were able to sprout ink, but not much, just one dot. And when most of them hatched, we released them back into the sea by the Cornish. So that was one memory I had when we were kids. So.
Brilliant.
Yeah.
Thank you for sharing.
You're welcome.
Thank you. All right. I see a question here. Yves?
So, if a shark is chasing a cuttlefish and a cuttlefish inks, will the shark be scared and then just go away and find something else, or will it still chase?
Is it a question for Julia at the back? Yeah?
Do you want to answer, Antoine?
Yeah, I got the answer. So that's the worst strategy for the cuttlefish to ink when they're a shark, because the shark is not chasing for the smell and with the eyes, he's just chasing with electromagnetic pulse. So the best behavior to adapt to get with a shark is what we call the freezing. So the cuttlefish will stay on the sediments, we try to bury, and we try to reduce the electromagnetic field and also the heartbeat. Like this, the shark, if he's like more than 60 centimeters from the cuttlefish, he won't know where the cuttlefish is. Sorry. If the cuttlefish try to ink, he will just feel the cuttlefish and will manage to catch the cuttlefish. So that's why also for the test, it was really nice to see how anxiety will just make the cuttlefish choose the wrong, let's say, behavior to escape to the shark.
Thank you.
Okay, thank you. Do we have any question on this side? Yes, here.
Hi, I just have two questions. First one is that, are they endangered? Because I know some animals all over the world are endangered. So are also cuttlefish endangered or not?
Yes, very. They have like a really big brain compared to the body size. They have neurons all spread in their bodies, so they are very intelligent. And like I said...
Sorry, endangered, not intelligent.
Oh, sorry.
But it's good to know that they're intelligent.
Well, the status of the most species of cuttlefish is they don't have enough data to know, because the species that we have more data is the common cuttlefish that Antoine uses, that is the European one in the Atlantic and the Mediterranean, and the giant one in the coast of Australia. So the rest, like we said, even here we don't know how is the population going. So it's like that around the world.
Okay, thank you. My second question is...
I can add something. The problem is, as you've seen, it's a really short life cycle species. So if you overfish the species for 2 years, you can really destroy the entire population because they don't have the time to renew. And it's really short, if you fish them all, then it's over and they can be really impacted also quickly. But on the other hand, you can also expect that they will adapt quickly as you have several generations. So that's the pro and the cons of the short life cycle.
Okay, thank you. Then my second question is that, let me say that maybe some of them are in the place where, say the environment is not clean, like they have plastics around in the ocean and all that. Since you mentioned that they are colorblind, do they make mistake maybe to eat those plastics or something? Or they know their food, maybe they can, I don't know, sense or smell it, I don't know. Or they might make a mistake and eat those plastics.
Well, they have a lot of techniques to detect the prey. It's not only vision. They have mechanoreceptors in their skin also. So I'm not saying that is not going, actually, I'm not sure, but I don't think will happen a lot. Do you know, Antoine?
I think, I'm the same point of view as you. It won't happen a lot, or it will be really a big mistake. But usually, as you say, it has really a lot of chemoreceptors. It catches it. I think it will just throw it away because it won't eat it anymore. But if they have plastic in their body, it's mainly due because of the plastic the prey ingested. So then it's in their body for their turn when they eat, for example, a small fish with some plastics or shrimps with some plastics and so on.
Thanks. Yeah. One question. Is there a specific species of cuttlefish that easy to train? Or?
Well, the ones that are bred in captivity are easier to train. The ones that you capture in the wild and bring them to captivity is a little bit harder. But I won't think that will be a difference with the species.
Thanks. At the back. Leila? Yeah.
Thank you for this talk. It was very insightful. My question is to everyone. So in Antoine's talk, he said that the cuttlefish use the left eye for anti-predation and the right eye for predation, so when they hunt. But is there any sign in their evolution that shows exactly why they use the left eye for anti-predation and the right eye for predation, or is it just like that?
I'm not a specialist on this. I won't be sure of what I'm saying, but it's like this for every species. Like most of us, there is a theory, for example, on which if you're playing tennis against a left-handed instead of a right-handed, you have more chance to lose because now you're not used to this and you might look with the wrong eye and so on. So it's like for us, why most of the people are right-handed and a little bit are left-handed. And it's kind of the same things, let's say, which is should be encoded in the genes, I think. But then I cannot go further on the explanation. I don't have any clues on how it's written or developed.
Yeah, I don't know either.
I then have a second question if none of you guys want to talk.
Yeah, go ahead.
So is there a limit to the adaptation of cuttlefish? So for example, to change in pressure or the environment or the pH levels?
Sorry, can you repeat because I couldn't
Is
hear?
there a limit to the level of adaptation that the cuttlefish can have to different characteristics in its environment?
So how fast can it adapt, I guess?
Well, depends of the stressor. What I can tell you to salinity is a real challenge for them. They don't do well with differences of salinity. High temperatures, as we have here, is one of the things that I would like to study here. So, next talk.
And we tried with the pH to go to some range of pH. And when we went below 7.4, we've seen metabolic depression and the individuals stopped growing and so on. So we had some development trouble. So if you go below this level, then you will have really huge consequences on the animals.
Thank you so much.
Thank you. All right. Yeah, well, we can.
Hi, hello. Yes, I've got an innovative type of question more, and it's related to the blindness or the color blindness of the animal. I mean, is there any benefit compared to human blindness in terms of the process that the animal uses, you know, to filter color? Well, you explained the process of how it filters, but is there any benefit in that process that could benefit humans in terms of color blindness? Do you understand my question?
No, I understand your question. Well, I don't know the answer to that, but what I can say, for example, is we already use a lot of technology that is developed because of the cuttlefish. For example, the papillae, the muscles that come, the bumps that come from the skin, now they are doing some manufacturing like tissues mimicking the biology of this animal that also do that. So why not?
Yeah. It's just something that rang when I listened to it, because I know someone that has color blindness issues. So yeah, just in correlation. Thank you very much.
Thank you. All right, maybe last question. Okay, we have 2 more questions. Okay, we'll come after.
Can record. Oh, did you get the first part?
Yes.
I'm
yes.
sorry. GPS devices can record longitude and latitude in different units. Do you actually specify for the user in which unit they should present this?
That's a great question. So we thought about it, and we thought that the decimal was the best way to do it. So we've given an example in decimals. So that's what we expect everyone to record.
Are you sure they know, or is the little star indicating please use decimal degrees? Otherwise it will be meters to kilometers off.
Okay.
Maybe you can specify that or highlight it even better.
Right.
To avoid.
Yeah, maybe I can look at that. I could have some kind of a guide or information over there, or the way to convert it. That's a great suggestion. Thank you. Thanks. And we had a question here, and then we will stop here for tonight.
All right. Thank you so much. I actually came very late. So I just wish to know, I'm a bit curious about the ecological importance of cuttlefish to our marine environment. If actually we are talking about this, then it means it's very, very important. I just wish to know. Thank you.
Cuttlefish are part of a food chain. They are predators for a lot of species like fish, crabs, and other cephalopods, but they're also the prey of a lot of species like seals, dolphins, and sharks. So you already know, if you came to the Shark Talk in the other month, we talked about a little bit about that. When you take predators, it will change all the dynamics of the animals that are in the food chain below. So if you remove cuttlefish, it will happen the same.
Yeah. Antoine, do you want to add to that?
I totally agree. The predator will miss food, and then the low little prey will just totally disturb the rest of the ecosystem. So, yeah.
Just a very important component, like all the components of the ecosystem, actually. Finding balance, this is it. All right, well, I think we will stop here. Thank you very much for coming tonight. Thank you to the speakers.